Everyone, welcome back to my class. So yesterday we learned about transport from the back to the cytosol, that is transport of protein from back to the cytosol. It occurs by a reversal of the usual process of import, which is known as reverse translocation, retrotranslocation, or it is termed as dislocation. Why this process happens is, if there is a protein which is misfolded, that will be transported back to the cytosol for the degradation. By this process, the misfolded soluble proteins in the lumen are translocated back to the cytosol. The process of transporting of proteins back to the cytosol happens from first binding of chaperone to the misfolded protein, then disulfide isomerase will bind, then lectin will bind to the oligosaccharide, then it is targeted to the protein translocator complex, from which the protein gets out of the lumen to the cytosol. After that, ubiquitin ligase will come and bind to that polypeptide chain, where ubiquitin molecules are inserted to the polypeptide chain. Then deglycosylation happens, that the precursor oligosaccharide is removed with the help of N-glycanase. After that, the polyubiquitin chain are targeted to the proteasome for the degradation. This is what happens in the transport of protein from the back to the cytosol. Then we discussed about insertion of membrane proteins into the membrane. Whichever membrane proteins which are to be inserted into the membrane or embedded into the membrane, that will be inserted by this flow chart, or flow from to Golgi to plasma membrane. Whichever proteins which are set to destined for plasma membrane, it will be transported to the plasma membrane. Which all proteins are transported? First one is transmembrane integral protein. Transmembrane integral protein are embedded in the membrane by hydrophobic region that spans the lipid bilayer. These membrane proteins have 4 topological classes, that is type 1, type 2, type 3 and type 4. Let us further discuss about this distinction of type 1, type 2, type 3 and type 4. Just consider this image. The blue color part you are seeing is extracellular, and green color part is the cytosol. Here you can see type 1, type 2, type 3 and type 4. So we will be discussing it individually in the coming slides. How this is inserted, this was also discussed in the previous class. The N-terminal signal sequence on the nascent type 1 protein initiates the translocation of the protein. N-terminus of the growing polypeptide enters the lumen of the Then the signal sequence is cleaved. Then the sequence of the non-polar amino acid, that is stop transfer signal, stops the transfer of the growing polypeptide. Next, this integral sequence moves laterally and becomes anchored in the lipid bilayer of the membrane. Translation continues until the C-terminus is achieved. After the C-terminus of protein chain is synthesized, it loops out on the cytosolic side, making the protein to embed inside the membrane. So now type 1. So in previous slide I had shown that which is extracellular and which is cytosol. Blue is extracellular and green is cytosol. Okay. First type of transmembrane integral protein is type 1, that is single pass transmembrane. Here you should make sure that single pass is having 1 unit, multi-pass is having more than 2 units interconnected. Here you can see the single pass transmembrane protein. They have hydrophilic N-terminal. Whenever NH2 is there, consider it as hydrophilic. Hydrophilic means NH2 is hydrophilic. Hydrophilic means they are water loving portion. It is in non-cytosolic, means it is in outside exoplasmic phase of the membrane. Hydrophilic C-terminal segment on the cytosolic phase. It is having cytosolic. The COOH part is having in the cytosolic phase. It contains internal stop transfer signal, also acts as a membrane anchor sequence and cleavable N-terminal signal sequence. They have both stop transfer signal and cleavable N-terminal signal sequence. Common example of type 1 protein are LDL receptor, insulin receptor, growth hormone receptor. Type 2, it is also a single pass transmembrane protein. They have the exact opposite of type 1. The COOH part is in the exoplasmic side and NH2, the hydrophilic part, is towards the cytosol. Have single non-cleavable internal signal sequence which acts as both start transfer signal and membrane anchor sequence, that is stop transfer sequence. Example of type 2 protein is transferrin receptor and Golgi galactosyl transferase. To correct one thing here, this has to be hydrophobic, means that is, this is by mistake, it is hydrophilic, it is hydrophobic. Wherever you see the C terminus, it is hydrophobic and it is not hydrophilic. Kindly apologize for this mistake. So this is hydrophobic. I'll write HB, hydrophobic, and the NH2 is hydrophilic. NH2 is hydrophilic. Kindly don't get confused. Here this has to be hydrophobic. Hydrophobic. Okay. Next. Type 3 is also a single pass transmembrane protein. They do not contain any cleavable signal sequence. They have N-terminal. What is N-terminal? I taught you right now. N-terminal is having NH2 and it is hydrophilic. Hydrophilic. It is on the non-cytosolic side. What did I taught? The blue color is exoplasmic phase. Exoplasmic phase, and also green color is cytosolic phase. Cytosolic phase. The N-terminal is on exoplasmic side and C-terminal is on cytosolic side. Have single non-cleavable internal signal sequence which acts as both start transfer signal and membrane anchor sequence. Example for this type is cytochrome P450. Then is type 4. It is a multi-pass transmembrane protein. You can see over here it has the multi-pass type of transmembrane protein. They have both N-terminal and C-terminal oriented towards the same side, or it may be the two ends have opposite orientation. Here you can see the opposite orientation. In some cases both NH2 and COOH can be found in the extracellular phase, or both COOH and NH2 can be found in the cytosolic side. If the type 4 protein has the even number of transmembrane helices, both its N-terminus and C-terminus will be oriented towards the same side of the membrane. If the type 4 protein has odd number of the transmembrane helices, both its N-terminus and C-terminus will be oriented towards the opposite side. Here you can see the odd number of transmembrane helices. Therefore, NH2 part is N-terminus towards outside and COOH is opposite to it. If there is another helices here present here, then there is a case of COOH and NH2 at the same phase. This protein contains many stop transfer sequences. Example of type 4 protein is beta adrenergic receptor, CFTR and GLUT1. Next, these are some examples of type 1, type 2, type 3 and type 4 multiple transmembrane helices. You can see these are the examples of different types of transmembrane integral protein of different topological classes.